joining us. I'm so happy to be doing this discussion with Ying. Ying Qian is the director of the SMA Foundation, and it seemed to me the perfect culmination for this series of master classes that we had done, because SMA, and in part because of the work the SMA Foundation has done, has had the good fortune of not only having a gene therapy, but an ASO and a small molecule all come to market. And I think this will give us a real world context to the discussions we've been having over the past 2 days. But maybe for openers, we can start with SMA itself, spinal muscular atrophy.
Yeah, actually, let me just grab the clicker here. So just a brief description of SMA as a disease. So it's a severe neurological disease, and even though it is actually a rare disease, it's a fairly common rare disease. If you think about it, 1 in 50 people are carriers of the recessive gene. And so 1 in about 10,000 children are born with SMA. And then it actually affects all people in racial groups somewhat evenly, and we used to call it the #1 genetic killer of children under the age of 2. That is no longer the case because we do have several therapies. It is also a neuromuscular disease, so it affects mostly the proximal muscles near the spine, and it really renders the kids sort of unable to move and ultimately unable to breathe. And the cognitive and emotional aspects are not really impacted. So SMA does vary quite a bit, the physical manifestation. If you look at the chart on the right, there's motor function, and then type 1, type 2, and type 3. We kind of roughly characterize them into these 3 or 4 types, I guess. Type 4 is really uncommon. And you'll see their motor function declines over time, but type 3 is significantly better. Type 3s usually we say are the walkers, type 2 are the sitters, and type 1 are extremely severe. And so SMA is caused by a deletion or a mutation of the SMN1 gene. And we need this gene to make SMN protein. And we all need SMN protein to keep our neurons and our muscles very healthy. Actually, now we're also discovering, as we're doing more research, that it is a whole body disease and that we actually need SMN in pretty much all of our cells. But when we first started, we were really focused on the neuron. But the reason why humans have SMA, but not like mice or monkeys, is because other animals do not have a backup SMN2 gene. So we happen to have this other gene that makes a small percentage of full-length SMN protein and a lot of SMN protein that is not full-length and not really functional and degrades very quickly. So through having that small amount, we are able to stay alive, whereas we actually do see SMN1 gene deletions in other animals, and they are not live-born. So that is SMA.
Yeah, I want to go back to the first slide.
Yeah.
You said it in such passing that, oh yeah, it used to be the #1 killer of kids under the age of 2, but no longer is. But I mean, it's a remarkable thing to say that because of the therapeutic development that's occurred and the fact that these treatments are now available, this is no longer the case.
Yeah.
What is the prognosis for today for a child born with SMA?
Yeah, the prognosis has changed so dramatically since the approval of 3 drugs. We go from seeing, more older patients having quite a bit more control over, say, their wheelchairs or being able to have a lot more strength in their hands or their arms, to kids who would have otherwise died who are now running, jumping, and playing on a playground like normal children. It ranges the gamut. There's a lot of different factors that go into how successful the treatments are for quote unquote curing the disease, but pretty much everyone has had some benefit in terms of their physical function.
Talk a little about the SMA Foundation itself and how it came about.
So the foundation was founded by Lauren Ang and Dinakar Singh. They have a child who at the time was 1.5 and diagnosed with SMA when a family friend noticed that she wasn't walking properly. They took her to a neurologist, and luckily, because they were in New York City and went to a very prominent neurologist, was actually diagnosed. So back then, actually diagnosis with SMA was really challenging. And then they started donating to other organizations for SMA, but they weren't really satisfied, I guess, with the fact that they weren't as much research focused and more like patient advocacy or care focused. And so they decided to start their own foundation and put all the money towards looking for a therapeutic treatment and not towards, so we don't really work on advocacy or patient care. Yeah.
And at the time they started doing that, what was the lay of the land for SMA research? How well understood was the condition, and what were the barriers to drug development?
Well, so at the time, there was some research going on at various universities. There wasn't a lot of funding, and there wasn't a ton of interest because it's a rare disease. And the landscape back 20-something years ago wasn't that everyone was interested in these rare diseases. Everyone was still very much focused on like cardiology and things like that. But there were some, and there was a patient registry, but it wasn't like a very detailed and scientific patient registry. It was like whoever wanted to register on the website could. A doctor could register you, a family member could register you could register yourself. We had people registered multiple times, and it was just like very voluntary and not very well known, I guess. So it was a bit of a helter-skelter, I guess, and there just wasn't quite as many researchers interested in it because of the amount of funding. Yeah.
You think about ways that patient groups can work to de-risk research, things like a natural history study, animal models. What existed in that regard, if anything, and where did the foundation put its early efforts?
So there was no natural history study, essentially, when the SMA Foundation was started. We made a huge effort and huge investment into putting that together. The animal models, there were some, but even to this day, we are still, I think we have maybe over 100 animal models, but we only use a few that we did eventually develop, and we are still actually developing it. The animal models. We do primarily use mice, and it's either too severe or not severe enough, and it's not great at mimicking the human experience. So the SMA Foundation did put a lot of effort into both developing animal models and natural history study, as well as other preclinical tools and clinical tools in order to facilitate research by companies.
The foundation started in 2003?
Yeah.
And by 2006, there was a relationship formed with PTC Therapeutics? I mean, that's remarkable. How did that happen?
Well, I think even from the very get-go, because Lauren and Dinniker were already fairly involved in the SMA field before 2003, they immediately started the foundation and started looking for relationships with pharma companies to try to develop a therapeutic. And through Chris Larson, who was an SMA researcher at the University of Missouri, and just talking to a lot of different people, they met various companies, including PTC. And PTC at the time was a little bit of a baby company themselves as well. And so they were enticed by the SMA Foundation saying, hey, we'll give you this, somewhat sizable pot of money. Eventually it was about a $14 million something investment. To help them de-risk their program and to work on SMA.
And what exactly did they do?
Well, they started screening. So they had a screening library, and they started screening compounds for SMA, where we've eventually found something that we thought was pretty good, and they started just working on the classic small molecule pathway of optimizing the chemistry.
So it always helps to be able to write multi-million-dollar checks to entice a researcher, a drug company, but
yeah.
It does take more than that. What was Lauren able to do to convince PTC to actually pursue this?
Well, Lauren and Dinger both come from finance backgrounds, and so they were very good at putting together slides showing like the financial aspects. So they approached it from a business. They were like, we can ask you to care, but why should you care about SMA? There's got to be some other reasons. Like there's so many diseases that need help, and so many diseases that are just heart-wrenching. SMA being one of them, but SMA, as you can see here, is actually fairly common for a rare disease. And looking at the prices that rare diseases research was sort of on the up at the time, and the drug pricing was starting to increase, and they just ran the numbers and said, hey, like if you develop this drug, this is a disease that kills people. Like everyone will be on it. Insurance companies will be forced to cover it, and you can make a lot of money. So.
And the fact that it was a genetic disease, I take it there was a case to be made there?
Yes, because it was easy to identify. Newborn screening was going to be possible. Otherwise, like diagnosis could be potentially an issue, right? But not so much in the case of SMA.
And you understood the mechanism of the disease?
Yes, and we understood the mechanism of the disease very well. Like we knew we were very lucky to understand that what causes 95% of SMA, which is when we talk about SMA, we are just talking about 5q SMA, which is what the three drugs treat. So we had a very good understanding of the underlying biology. So that may or may not be step number one, I would say, for a lot of people. I think you do need to understand the underlying biology, but sometimes the SMA Foundation started pushing forward without understanding all of the underlying biology before attempting to create a drug. But you do have to know, like, well, why, what you're shooting for, I guess. Yeah.
Were there lessons learned from working with PTC and others in terms of how a patient advocacy group could build relationships with pharmaceutical companies?
Yeah, I would definitely say so, because we really wanted to be partners with them in terms of working on the drug together, because we had a deep understanding of SMA. We wanted to drive the timelines because Lauren and Dinegar's child was getting older and getting progressively worse. And so we wanted to be partners with them in the development of the drug.
And how receptive were pharmaceutical companies to having patients as partners?
I think if we were just patients, it might have been challenging. However, everyone who was working at the foundation at the time had come from a drug development background, fairly high-level drug developers, I would say. And so, they knew the talk, they knew how pharmaceutical companies worked, and they could really communicate with them as equals. And also, we were providing a lot of the, tools necessary for drug development.
That means cash.
We had paid for all of those studies, and providing those to them for free.
So you mentioned the staffing. If you go onto the SMA Foundation's website and look at the team, you'll see people with titles like Chief Medical Officer and Chief Clinical Officer and Research Strategist. And this is not a big organization.
No.
How many people are on the team?
Well, there's 3 paid employees, actually, and then we have chief medical officers. They're actually volunteers.
But what's the thinking in terms of, it looks very different than a lot of small patient organizations. So
yes. What's
the thinking in terms of how you staff and what the organization is geared for?
I mean, we call ourselves a virtual biotech or pharma. We really staffed for people with deep experience and in drug development. And that's really what we were looking for, because we are interested in developing a drug. And so people's backgrounds are always coming from pharma. They're people who have put other drugs through the pipeline and been successful at developing drugs. So that was the kind of expertise we're looking for. And we did hire those people initially. We were a larger organization back then, but we've had the good fortune and success. So now we do actually have volunteers.
So talk about Karen. Who is she and where did she come from?
Karen came from Roche Genentech. She worked a long time in drug development, and she was also in neurosciences always. So in the same field, in drug development, she knew a lot of, Karen's also, if you've ever met her, is particularly social. So she's very good at talking to everyone. Sorry, you can't have Karen. But she's very good at talking to everyone and making connections. So we've also been very helpful. I think one of the things we've done is that we haven't just been solely focused on our mission. We've also been quite helpful to other organizations and connecting them. So the good karma comes back to us, I would say sometimes, and then we're able to make all those connections with various pharma companies.
We should say Karen is your CEO.
Yes, Karen's our CEO, yes.
So PTC took this drug that was in development and they partnered with Genentech. Was that facilitated by Karen? Was it a coincidence that Genentech ultimately took over the development of this drug?
I would say it was not facilitated by Karen. We also have a unique, I guess, advantage in that Dinakar Singh works in pharma. He has a hedge fund that invests in pharma and clinical trials. So he also knew a lot of the people on the finance side in pharma. And so I would say the partnering, the business relationship came more from that side.
So if you look at the way biopharmaceutical companies talk, they'll always talk about patients and how patient-centric they are and how they partner with patients. Yeah. I've never seen a true partnership with a patient group the way PTC and Genentech did, where the SMA Foundation was at the table discussing clinical trials all the time. And not only that, it played an unusual role in that when there was a dispute between PTC and Genentech, it was the arbitrator and oftentimes the deciding vote.
Yes, it's a three-way partnership. And it is a unique situation, but I think we definitely had to earn it. We had been working with PTC for a long time before Roche and Genentech came and basically purchased the program. Not fully purchased, but partnered into the program. And so they realized that PTC really respected us and that we were developing, hand in hand with them through that whole process. And they were just arriving at the sort of tail end of that process. And we had we had to still prove ourselves again that we had good input. We did a lot of work into their clinical trials. Like I personally had to go to some of the clinical trial sites and talk to them because for example, in China that I had some connections there, I went and they didn't have the correct list of children's hospitals because they didn't have a deep local knowledge. And then I was able to make some phone calls and get them into the hospitals. So there's various things that we've done to help them along the process that has helped us earn our seat at the table. But now I would say it's a very strong collaboration. Like we're concerned about some SMN in the bone. And so, we recently had a meeting with them and said, hey, could you look at the efficacy or the penetration of Evrysdi into the bone? These are things that they now listen to us and will look into on our behalf. Not really on our behalf, it's really for all of us. But yeah, we had to earn that.
So I've had a tendency to ask the drug developers on the stage about what patient organizations can learn to work with them, but what do you think drug developers can learn from this experience and how they should be working with patient organization groups and how they might better value the input they can provide?
I think that as much as you can learn as much as a drug developer can learn about the disease on paper, it's a different experience to talk to a patient. And when the patient they can give you valuable feedback on what kind of outcome measures and how to design your clinical trial. I think that's probably one of the most critical parts because to really understand the nuances is very challenging. And to work with the patient and the doctor together to be like, well, really because in a clinical trial, it's such a limited amount of time and it's so hard to really find a measure that you can really test. Like, patients will tell you it's efficacious, but you have to show it in a really limited amount of time. Anesthesia and their respiratory function can really be compromised during the procedure and after. So I just think it's very challenging for people to undergo general anesthesia every 4 months. But not everyone has to have that, and not everyone doesn't tolerate it well.
And this was an expensive therapy. I think $750,000 for the first year, or
the
cost of half of that.
Yeah, I honestly can't. It's about that price. Yes.
Big dollars. What?
Big dollars. Yeah.
How difficult is it to get a payer to pay for it?
It hasn't been difficult, I would say, in the U.S. to get a payer to pay for one of these therapies. Depending on the payer, they may say that you need to be on a different therapy, although they are pretty much around the same price. I mean, Evrysdi and Spinraza are both around the same price. Evrysdi is a little bit cheaper and doesn't come with the associated hospital costs, because the $750 that you're mentioning is really just the drug cost, not even the intrathecal injection cost. But it can be very challenging in other countries, both from a technology standpoint and from a payer standpoint. But in the US, it is one of these 3 therapies, or all 3 are widely covered.
And people around the globe have this disease, so if you need access to hospitals and drug and procedures, interventional radiologists.
I mean, some places it's not even possible to get a lumbar puncture with fluoroscopy. It's just not going to be possible to do that, especially with a patient who has difficulty traveling as well. It's hard for them to fly. It's hard for them to get on a train or a car, and long duration travel is just very, very challenging. So, not only do some countries not have it, but patients may not live that close to a major center. Yeah.
Zolgensma was next. This is the gene therapy.
Yes, Zolgensma was next.
How does Zolgensma work? How is it administered?
It is administered to only patients under the age of 2 because of the complications with receiving AAV9.
Which is the viral vector.
Yeah, the viral vector. They test, obviously, the patient first for a previous episode of disease, because if you have had that, it can kill you. And as was mentioned in the previous session, 2 patients have actually died, one in Russia and one in Kazakhstan, from acute liver failure due to receiving the virus. But it is intravenously injected. They are currently in trials for intrathecal injection to older patients, because the reason why they don't do it after the age of 2 is because you are too large. You have too much body mass and require too much AAV9 to safely do it systemically, as well as increased likelihood of prior AAV9 infection.
And this is about $2.5 million a dose? Is that something like that?
I thought it was one something, but
okay.
But it's really expensive.
We're talking about a 7-digit.
Yeah, it's a really, really expensive. It is one time. However, as mentioned in the previous session, one time doesn't mean, oh, I'm cured, I'm good. Because we do not want the virus to replicate, it's not CRISPR technology, right? It's the gene therapy technology, so that it introduces a piece of the SMN1 DNA through the virus into the cell nucleus, but not into the DNA. So it will not replicate with your cells, nor do they want the AAV9 to replicate, because then you have unchecked viral replication, right? So basically what you get and what goes into your cells when you first get it is all you have. And when those cells turn over and die, like in the case of muscle cells, it's gone. So neurons don't, yeah.
So this is what people talk about, durability.
Yes.
How long lasting is it? What do we know about the durability of Zolgensma?
So I guess SMN protein is most necessary at the beginning of life. So we haven't really, I think, gone back and, maybe Novartis has gone back and looked at it, but we do not know about the durability in all the cells. All we know is that because neurons don't replace themselves, right? So the durability of AAV is probably pretty good at a neuron. but it's not good in a muscle and other cells as the child grows. But because it is most necessary at the beginning of life, the outcomes have been pretty good.
So when we were talking about PTC Therapeutics and its partnership with Genentech, that resulted in the drug Evrysdi, which is a small molecule drug. Yeah. Do we know how Evrysdi works?
Yeah, it works very similar to the ASO, but unlike the ASO, which is large, it is small. So it's an oral drug, and it gets through the whole body. And it also attaches to the pre-mRNA and helps the splicing to create the full-length protein.
So what's it like to use that drug if you're a patient?
I mean, it's orally every day. You can take it with you. It's a liquid formulation. They're still working on a pill formulation because that would be easier, because the liquid has to be kept refrigerated. But most of all, you don't have to go to the hospital. You just get it to your house, and you have to stick it in your refrigerator, which, is still a challenge for the entire world to have that the refrigeration capacity. But I would say, it's very different than having to go to the hospital every 4 months.
The cost is comparable to the ASO, you said?
It is comparable to the ASO.
Any issues with payers on getting that covered?
No, not typically in the US and in Western Europe. Still I think there's a lot of negotiations in the rest of the world.
So I alluded to this in our previous conversation, but if you think about a foundation that's seen not one, but 3 drugs and 3 different modalities come to market, you'd think you can hang up your boots and take the day off.
My parents would ask me, how come you're still working at the foundation? I thought you were done.
But the problem is that these drugs don't restore your abilities. They stop the disease in its tracks.
Yes, they stop the disease in its tracks.
So for a progressive neuromuscular condition, what does that mean?
So it means that babies who are born today have a pretty good chance, because even if you did get gene therapy at birth or one of the other ones, it doesn't mean that you are cured. People respond differently to every drug. And there's a lot of modifiers that we still do not understand as to how the outcome will be. But for a lot of patients who are alive today, they had more advanced disease. They were already in a wheelchair. They already had scoliosis. Those kids who are in a wheelchair are not going to get up and walk. They will have better function. Like, one of the patients we know was having trouble feeding herself, but now she can take things out of the microwave, pick up a glass of water, and these are huge differences. Now, it may not be incredibly captured on any scale. I don't know if other people would jump up and say, hooray, she's cured, but to her, it makes a big difference. However, as the SMA Foundation, we just feel like we could give them more. Also because we had gotten a whole bunch of money back from our investments in the other therapies. So we're not done. Of course, we understand that the problem we're tackling now is much broader, not as well defined as, replace a gene or a protein that we know doesn't exist or is missing. But now, how do we regenerate exons or axons? How do we regenerate those and muscles cells? And how do we get satellite cells to come out of quiescence? How do we make them strong? How do we get that connection going? How do we actually generate new neurons? These are all the questions that we're asking, because we aren't able to rescue or grow new neurons just from having SMN. It just rescues the ones that were dying off and makes them a bit stronger, but doesn't change what previously happened.
So the foundation has learned a lot about working with researchers and drug companies.
Yeah.
Having done this for 20 years, how is that shaping the approach you're taking on these regenerative therapies?
Well, we now have a lot of advisors, I would say. We've been very lucky that you know, we're not that many people and we don't know a lot, but we have a lot of friends who are willing to help us. So, and we're not really shy about asking for help and asking for their expertise. So we have put out to major universities and other researchers, be like, hey, your research looks interesting, because people tell us about their research, and then we say, oh, their research looks interesting. How do you think it can apply to SMA? Because we think it may be possible, but we know SMA and you know your research, and let's get together and talk about this. So, yeah.
So let's bring it back to modalities for a moment. When you're funding research, do you think about modalities? Is that a relevant thing for a patient advocacy group to even consider?
I think we only thought about how can we get a drug as quickly as possible. It didn't matter how it was going to be. You know, I think a lot of people do like gene therapy a lot because I think or gene editing even seems more promising in terms of being a cure. However, the foundation at the time in gene therapy, there are serious risks with gene therapy. So, yeah, there are things to consider.
So you mentioned kind of in passing that you now have money to reinvest from the success of these drugs. There's been some very big wins from the venture philanthropy side. You think of Cystic Fibrosis Foundation, Cure Duchenne. What advice would you give a patient advocacy group about whether to seek equity in a program they fund?
I would say definitely try to seek equity. We made a lot of sucker deals. We look like we got a lot of money back, and we do. It is a large number. However, it is a very small percentage. We could have probably written a much more aggressive deal, but that just wasn't what we were shooting for. So I think number one, you know, remember you're in there to cure your disease, and if you can get some money back, get it back, because you'll want that money later to fund further research. You're unlikely to be done, just like we are not done at this time.
Do you have any sense that a biopharmaceutical company would treat a patient advocacy group that's an investor rather than a patient group differently? Would they think of them differently? Would they talk to them differently?
I mean, I'm not sure. I don't necessarily think so, because I imagine the primary motivation for any patient advocacy group, even if they are the patient themselves, they care so much about, more than anything else, more than money, to care about treating the disease, right? So I don't think it's going to be that different if they ask for some money back versus not, because the money back is only on the success, based on the success of the drug. It's not any time before that. So, and I don't think any group would be more or less invested whether they were going to get money back or not. Now, of course, I would say one thing to be maybe mindful of is that you still need to be impartial. I think they might dislike it or not trust you if you were like, well, I have invested in this company and I get money back from that company, but not from this one. So they might think you're trying to help. So we try to be very, very, very neutral, just because we are a patient organization. I'm not a patient research organization. We are looking for a cure. We don't care if it's Zolgensma, from which we got nothing. And we still help them because it was still important to us that, a cure was being developed. We're not in the position to be the original funders for Zolgensma. However, I do caution that you do need to be really impartial on that, because you never know which company is going to actually have the successful drug, and what you want is a successful drug.
So I know you think about the unique situation of the SMA Foundation to some extent, that this was a couple that had the benefit of having a lot of access to money, that was able to attract someone out of industry to lead the organization, someone with deep drug development experience. But what lessons can other patient advocacy groups take from SMA's experience, and how they can help build relationships with biopharma, help get drugs into development, and also keep a voice in the development plan?
Yeah, I think the SMA Foundation has been uniquely lucky. However, I do think that there are people in pharma who want to work for a disease organization. It's just, I think the joy of working for a foundation or a disease organization is so great, and the responsibility and the personal drive is so great that I think you'll be surprised that you might find someone who has been in pharma for a while, who has decided that they perhaps want to leave, and that you shouldn't give up trying to seek that, because that person probably has a lot of experience. And I mean, granted, you're probably not going to get them for super cheap, just because pharma salaries are pretty good, but it doesn't have to be quite that level. And having met a lot of people who are volunteering for us, I do think there are a lot of people out there who really want to do good. And they may not have the disease, they may not know anyone with a disease, but the impact that they'll be able to make, and they know that they'll be able to do that, will help drive their motivation and helping you on your quest. So don't give up looking for that.
So as we get ready to take a few questions from the audience, Carmen has a mic. If you want to ask a question, just raise your hand. But as she does that, what one takeaway would you want people from this discussion to think about and take with them?
I think if you want a drug, you have to be really focused on drug development. There are a lot of patient advocacy groups, and I do feel that's very valuable. Patients need to have resources. They need to be connected. However, that takes away from, for us, it took away from, would have taken away from our sole mission to develop a drug. And so even though it seems rather heartless for us when we get letters, oh, could you help so-and-so? We actually say no. There's another organization that does that. We are truly just a drug research organization, and that is what we're focused on. So don't forget your focus, don't get distracted. Really hone in on what you want.
Any questions?
I apologize for my ignorance on SMA, but does the disease, are there a wide group of variants within the population, and so the therapies that you have developed, are they as effective generally across the population, or is there a lot of variability in terms of the effectiveness of the therapies that you've developed?
There is variability, but so the variation in SMA is based on how many SMN2 copies you have naturally. However, and so the two therapies that work on latching on to SMN2, what the SMN2 makes, it's going to be more successful for people with greater copies of SMN2 because they've got more to work with. So yes, there is some variability. However, the gene therapy is not influenced by this. However, there are so many modifiers that we're finding. Sometimes our copy number to type chart is not like a great linear graph. So there's still so much we don't understand. And honestly, I would say that's another takeaway. Don't try to understand everything about your disease before you go for the drug.
And there's SMA type 1, type 2, type 3.
Yeah.
Are they basically differentiated by time of onset or
Yeah, they're differentiated by time of onset and also by highest milestone achieved. But we don't even really use that terminology anymore just because the disease natural history is so different in the US now with that. It's like we usually go by copy number now just because like that is more predictive of how severe the disease might have been. And yeah.
So we've got to close. If you do have questions for
Just
Ying.
one quick question. One quick question. Thank you so much for the presentation. It was amazing.
Thank you.
And congrats on all the progress. Super inspiring. You're the research arm. There's other support organizations. I'm assuming the support organizations played a large role in enrollment of the trials because you guys were on the research side and were able to negotiate, right, percentage. You were able to negotiate with the success of the drug, getting funding back, you probably invested money. Was there any consideration for the organization that was on the support side doing a lot of the work on enrollment? Did they benefit at all financially from these wins?
So we were the ones who enrolled the trials because we are the ones who paid for the natural history study. And the natural history study patients were already accustomed to the clinical outcome measures being used in a clinical trial. So not only did we find the sites, we helped identify the patients through the natural history study that we paid for like 15 years before the trial even started. So actually they had very little to do with the enrollment, but they did benefit because they eventually did they did the same thing that we did. And one of the other large SMA organization called Cure SMA is getting money from Zolgensma because they funded Zolgensma. So they've done actually the same thing. Yeah.
Well, please join me in thanking Ying for a wonderful time.
Thank you.